Okay, I'm Einar Pontén. I'm CEO of Lipum. That is a biopharmaceutical company located in Umeå. Yeah, I know we are in Skåne now. It's 1,200 kilometers north. And why do I travel this far then? Yeah, because I have something important to tell you about, and that is that Lipum has identified a novel target molecule for treatment of chronic inflammatory diseases. And it goes back from academic research by the founders in pediatrics, where they actually discovered this protein as a key enzyme in fat digestion, in nutrition of newborn children and infants from breast milk. When they found this target molecule, the BSSL molecule in breast milk, they also discovered that it was present in the circulation, in urine, in blood. And they tried to figure out where it was formed. So in that search, they explored plaque and also liver samples. And in liver samples that were from patients having fatty liver, they found very high levels of the BSSL molecule. And fatty liver is an inflammatory condition. So that was the starting point for looking into more details of this. They figured, if you don't have this protein, will you then have less inflammation? And to first examine this, the knockouts, the mouse that were deficient from the protein were exposed to a well-established animal model, the collagen-induced arthritis that is used for rheumatoid arthritis. And as you can see on the right-hand side, that the controls, when they are exposed to this model, they develop the disease as expected, while the deficient mice, they are more or less unaffected. And another type of chronic inflammatory diseases are inflammatory bowel disease, like Crohn's disease and ulcerative colitis. And the model for that is the DSS-induced colitis. And in the same type of experiments, the knockouts, the colon of the knockouts that you can see on this image are almost unaffected, while the control group are seriously affected. So there is an interest to explore what happens then if we block the activity of the BSSL by using anti-BSSL antibody. And this was tested in another model, the Pristane-induced arthritis model on rats. And regardless of the route of administration of the experimental antibody, the animals were considerably less affected compared to the control. And that is also very well illustrated by these photos. And to explain the mechanism of action, it was by various studies found that the BSSL is released from the neutrophils. And when it binds to the monocytes, the white blood cells, they are activated and migrate to the site of inflammation and also differentiates and stimulate and maintain the inflammation. So the therapeutic idea is then to block the BSSL molecule by an antibody, and that then becomes the product. And luckily for Lipum, a couple of years ago, the researchers were supported by SciLife Lab. You know, SciLife Lab is a national resource for molecular biosciences in Stockholm-Uppsala area. And that project was then to develop and optimize a therapeutic antibody for blocking the BSSL. And starting then with around about 17 experimental antibodies, merging them with the strategies of antibody development and the libraries at the SciLife Lab, that ended up with 38 IgG4 antibodies that were further examined and evaluated. And in the end, that was decreased, the number were decreased to 5 antibodies. And these were actually also put into cell line development to test the manufacturability. And out of these, there were a candidate drug, a lead candidate drug selected in July this year. And the cell line development is such a state that we will start the production development within a month from now. And that means that we are a preclinical stage company, but we are very rapidly approaching tox studies. We will have non-GMP material this spring. We can do the tox studies, and in the end of next year, we will have GMP material and can start the clinical program to get the clinical proof of concept by end of 2022. So what is the indication? Then, as I mentioned, the founders of the company have their background in pediatrics, and they have for very long been well aware of the medical need within the field of juvenile idiopathic arthritis that affects children. That is a rare disease. Just about 300,000 children in the age up to 16 years old are affected. Beside the stiffness, the pain they have, they also refuse to walk, and they want to stay home from school. So that disease also has social consequences and high costs for the society. The current treatment are, in the third line, typical anti-TNF drugs. But as you know, in many cases, there are non-responders that actually have no effect of anti-TNF drugs. So they end up in the fourth line treatment where there are very few therapeutic alternatives. And altogether, it's around about 80,000 patients. We aim for an orphan drug designation, and we have estimated that the addressable market or the possible market for Lipum is around about 800 million euro. So our current status is that we have a very comprehensive preclinical data material on rheumatoid arthritis, but also on the IBD field. We have a fully humanized monoclonal antibody candidate drug selected soon in production development. We have received financing support from Horizon 2020. We are in the middle of that project, and we raised 2.4 million euro in a private placement in March this year. What we focus on now is to extend the preclinical program to explore more indications, because we believe that an anti-BSSL antibody will have a wide scope. And that is, of course, attractive because there are a number of inflammatory diseases, chronic inflammatory diseases that have a high unmet medical need. So first of all, we aim for an orphan drug designation, and we estimate that we will need round about 18 million euro to get to proof of concept in 2022. And the next financing round will be during the first half of next year, especially in particular to finance then the GMP manufacturing. I think I was faster than 10 minutes, but someone should be that, so I thank you for your attention.